and welcome back to my channel. So today's video is my final immunology video, and this is for OCR A Level, both OCR A and OCR B. So again, this video is going to be a bit of a long one. OCR love immunology questions, and so I've included all of the immunology questions I could find. So there's a lot of questions at the end. So I'm just going to quickly sort of go through the main ideas of the specification points. So for OCR A, it's non-specific defences, the structure and action of phagocytes, also the structure and roles of B and T cells, as well as the primary, secondary immune responses and antibodies. And then for OCR B, we again have primary and non-specific defences, phagocytes, roles of B and T cells, the secondary immune response and antibodies as well. So I'm just going to discuss pathogens. So a pathogen is any organism that causes a disease. So some of the main examples you know are probably bacteria and viruses. And disease is defined as a condition that impairs the normal functioning of an organism. And both plants and animals can get diseases. So bacteria, so they're prokaryotic cells, they don't require a host to survive, and they contain structures such as a cell wall, membrane and cytoplasm. A virus is described as a microorganism but not a cell. It is a nucleic acid surrounded by protein, and they do require a host to survive. They do not possess the structures that bacteria do. So some of the primary non-specific defences you need to know about for biology A are the skin. So the skin is a physical barrier, and it contains a protein called keratin, and it's this protein that blocks pathogens from entering the body. It also has a chemical barrier, so it produces sebum, which is an oily substance, and this prevents pathogen growth on the surface of the skin. And also it has something called skin flora. So this is a microbial barrier, so they are healthy microbes that outcompete pathogens to space and nutrients on the surface of the skin. Another non-specific defence are mucous membranes. So these line the body tracts, so including gastric change system airways, so the respiratory system, and they secrete mucus. And mucus is a sticky substance, so it's able to trap pathogens, and it also contains enzymes called lysozymes, which destroy bacteria and fungi by breaking down their cell walls. Mucus also contains phagocytes, which are a type of white blood cell, and these remove any remaining pathogens. We then have blood clotting and wound repair. So we have platelets, which are really tiny blood cells that help the body to form clots, and they come into contact with collagen, which is a protein in the skin, or the damaged blood vessel wall, and what happens is they stick or they adhere to the wall and they secrete several substances. So these substances are thromboplastin. Now this is an enzyme which triggers a cascade of reactions, so it causes a blood clot to form, which is also known as a thrombus. So platelets also secrete serotonin. You've probably heard of this as the happy hormone, but this plays a role in making the smooth muscle vessel walls contract. So this causes the walls to narrow and to reduce the blood supply to the area. So then the clot dries and it forms a scab, and this scab is what keeps pathogen out. And then we've got epidermal cells. So these are below the scab, and these are what make up the skin. So these develop and they regrow around and within the damaged blood vessels, and then once they have fully grown, collagen fibres are deposited to give the tissue its new strength. So then we have expulsive reflexes. So this includes coughing and sneezing. So the reason we do this is to get rid of pathogens, and we do this by releasing mucus, and also vomiting and diarrhea as well. They help get rid of infective pathogens. So then for biology B, we have the stomach acid. So the stomach produces hydrochloric acid, which is very acidic. It's pH of 2. And this is what kills the majority of pathogens that enter via the mouth and travel down the esophagus as a part of the digestive system. We then have mucus and cilia in the respiratory tract. So we discussed mucus previously. So this is what traps pathogens, and it also contains antimicrobial enzymes. We then have cilia. So cilia are hair-like structures, and they beat in waves. They push the mucus up out of the respiratory tract in order to be coughed out or swallowed to then be destroyed in the stomach. We then have lysozyme enzyme. So mucosal surfaces such as the eyes, mouth and nose all produce secretions. So we have tears, saliva and mucus. So these secretions contain the enzyme lysozyme, and the lysozyme kills bacteria by damaging their cell walls. So it causes bacteria to burst, which is known as bacterial lysis. So for both specifications, you need to know about inflammation. So this is a localized response to pathogens, but also to irritants or damage as well. And this results in inflammation at the site of a wound. So it's characterized by pain, heat, redness, and also tissue swelling. So the process of inflammation begins. So the chemical histamine causes vasodilation, which is the widening of blood cells. So this vasodilation causes the localized heat and the redness of the skin. So this increased temperature helps to prevent pathogens from reproducing or replicating. It also causes blood vessel walls to become more leaky. So this means that they open up more, so blood plasma is forced out into the tissue fluid, causing edema, which is swelling. The role of cytokines is to attract phagocytes to the site of inflammation or the site of infection and to dispose of the pathogens by phagocytosis. And we'll discuss the process of phagocytosis in more detail in a little while. So there are several types of white blood cells. So the ones that we're going to discuss are phagocytes to start with. So these are macrophages and neutrophils. So these are some examples of phagocytes. So the overall role of them is to engulf and destroy pathogens. So they are cells that build up at the site of infection and attack pathogens. So specifically, neutrophils are involved in ingesting and destroying bacteria. So if you have a cut or a wound, you can often see pus, and pus consists of dead neutrophils and pathogens. So then we have macrophages, so they engulf bacteria, foreign matter and cell debris. So now we're going to discuss the process of phagocytosis. So this is performed by both neutrophils and macrophages. So to begin with, pathogens release chemicals attracting phagocytes to the site of infection. And then the phagocytes recognize the non-human or non-self proteins, which we discussed earlier, which are known as antigens, which are present on the pathogen. The phagocyte then engulfs the pathogen and it encloses it in a vacuole, and this vacuole is known as a phagosome. Another organelle known as a lysosome then fuses with this phagosome and it exposes the pathogen to lysozyme, which is the hydrolytic enzyme that we spoke about earlier. So lysosomes contain lysozyme. We then have the lysozyme from the lysosome digesting and destroying the pathogen. And then finally, the phagocyte absorbs this pathogen and the pathogen's antigens combine with an MHC in the cytoplasm. So MHC stands for major histocompatibility complex. And this is basically an antigen complex which is displayed on the phagocyte membrane, which makes it an antigen presenting cell, which allows other white blood cells to recognize it. So in terms of the roles of neutrophils and macrophages, neutrophils arrive first. So these engulf and destroy the bacterium in under 10 minutes. So they usually destroy between 5 and 20 bacteria, and they consist of a lobe nucleus and granules in the cytoplasm. Macrophages, which are longer lived and larger, they take longer, but they undergo a more complex process. So they can destroy up to 100 bacteria. So it begins by a macrophage digesting the pathogen, which then combines with glycoproteins on the surface membrane called the MHC, which we discussed. The MHC complex then moves the pathogen antigens to the surface, making it an antigen presenting cell. So some important chemicals involved in phagocytosis are cytokines and opsonins. So cytokines are produced by phagocytes once they have engulfed a pathogen. So cytokines are cell signaling molecules, so they inform other phagocytes that the body is infected, and this stimulates them to move to the site of infection or the site of inflammation. So cytokines increase body temperature and they stimulate the specific immune system. We then have opsonins. So these are chemicals that bind to the specific pathogen and they act as a tag, so they're more easily recognized by phagocytes. The phagocytes have receptors on their cell membranes that bind to these opsonins and then allows the phagocyte to engulf the pathogen. There's lots of different opsonins, but antibodies such as IgG and IgM have the strongest effect. So as I said previously, we are going to discuss antigens. So antigens are a specific molecule, a protein molecule that's on the surface of all cells. So these are specific to the specific cell. So the body recognises the difference between self-antigens and non-self antigens, the non-self antigens being, of course, on pathogenic cells. And some toxins also act as antigens. So antigens trigger an immune response, and the immune response involves the production of polypeptides, which we call antibodies. So the specific immune response is initially slower than non-specific responses and can take up to 14 days to respond effectively to an invading pathogen. So this is known as the primary immune response, and this is when we are first exposed to a pathogen. Now, the good thing about the immune system is it has memory cells. So if we're infected by a pathogen again, the body can react very quickly and destroy the pathogen, usually before we experience any symptoms. So we're going to discuss the structure of antibodies. So these are Y-shaped glycoproteins and they are immunoglobulins. So they have 4 polypeptide chains, so 2 are heavy and 2 are light. These chains are held together by bonds known as disulfide bridges, and the bridges themselves also hold together polypeptide chains. And then we've got the antigen-antibody complex. So this complex is formed when the antibodies bind to the antigen, and this is a specific antigen. So antibodies are specific to a unique antigen. It's like a lock and key mechanism similar to enzymes and substrates. So antibodies also have a variable region. So as you can see in the name, this means that it changes based on the antigen. So the variable region consists of 110 amino acids on the heavy and light chains, and it is a binding site. So there's a different shape for each antibody, which is what gives the antibody its specificity. The antibody also has a constant region, so this stays the same in all antibodies, and it also has a hinge region. So this region provides flexibility, so it allows distance between the binding sites to vary, which means that the antibody can bind 2 separate antigens at each of its antigen binding sites. So you can see the antibody structure more clearly here. So you can see our antigen binding sites, you can see our light chains, our heavy chains, and also our disulfide bonds or our disulfide bridges, as well as our hinge region and our receptor binding site. Now, OCR love to give questions about antibody structure. They usually include perhaps a half-completed antibody diagram, or ask you to make sure you know what the labels are on them. So make sure you are familiar with this structure. So antibodies act as many different things in order to defend the body. So they act as opsonins, which we discussed previously as chemicals involved in phagocytosis. So antibodies acting as opsonins allow antigen-antibody complex to be easily engulfed and digested by these phagocytes. They also act as agglutinins, and these are what allows the antibody-antigen complexes to clump together. So this prevents them spreading through the body and allows the phagocytes to engulf several pathogens. Antibodies also act as anti-toxins, so they bind to toxins produced by pathogens and they turn them into a chemical which is harmless. We're now going to discuss this specific immune response, and this involves B and T lymphocytes, which are more colloquially known as B and T cells. So B cells and T cells are both created by bone marrow stem cells. The difference is B cells are mature in the bone marrow and T cells mature in the thymus gland, hence B cells for bone marrow and T cells for thymus. So the B lymphocytes are generally involved in the humoral response, and T cells are generally involved in the cellular or the cell-mediated response. B lymphocytes can differentiate into memory cells and also effector cells, as well as plasma cells, and T lymphocytes differentiate into memory, killer, and helper T cells. So there are several different T cell types. So the first we're going to discuss are T helper cells. So CD4 receptors on the cell surface membrane are what bind to surface antigen on antigen presenting cells. So T helper cells have receptors called CD4 receptors. T helper cells also produce interleukins, and these are a type of cytokine. And these interleukins stimulate B cell activity. And this stimulation allows antibody production to increase. It also stimulates production of other T cell types, as well as attracting and stimulating macrophages in order to ingest pathogens with antigen-antibody complexes during phagocytosis. We then have T memory cells. So these are involved in the secondary immune response. So they're able to live for a long time. And when you're exposed to an antigen on a pathogen for a second time or a third time, they rapidly divide to form lots of clones of T killer cells which can destroy the pathogen. So the T killer cells destroy the pathogen carrying the antigen, and they do this by producing a chemical called perforin. So perforin produces holes or pores in the cell membrane of body cells, allowing them to become freely permeable, and this is what kills the pathogen. So finally, we have T regulator cells. So these are very important in suppressing the immune system in order to control and regulate it. And you may think, why do we want the immune system to be suppressed? Well, we want to make sure that the immune response stops once the pathogen is eliminated. Otherwise, this can create an autoimmune response, and this involves the body tackling our own cells and destroying them, which of course we do not want. We need to ensure the body recognises self-antigens, and interleukins are also important in this control. We then have B cell types. So B effector cells are important in differentiating to produce plasma cells. So we know plasma cells are the cells that produce antibodies which are complementary to a specific antigen, and the plasma cells release these antibodies into circulation in the immune system. And active plasma cells, they only live for a few days, but they produce more than 2,000 antibodies per second while they are alive and active. We then have B memory cells. So like T memory cells, these live longer, so they can remain for months or years in the body, and they enable the individual to respond more quickly to the same antigen in the future. So this, like T memory cells, is part of the secondary immune response. So I've briefly gone over the primary and secondary immune response, but I'm just going to discuss in a bit more detail here. So the primary response is the first exposure to pathogens. So the first time a B cell comes across a non-self antigen that is complementary to its cell surface receptors, this causes the production of sufficient antibody producing cells, and this can take between 10 and 17 days. So due to this taking time to produce antibodies, symptoms are suffered. We then have the secondary response. So if you're reinfected by the same pathogen, so second exposure, third, fourth, fifth, any exposure that isn't the first exposure, this causes B memory cells to immediately differentiate to produce plasma cells which release antibodies. So then the pathogen is able to be destroyed before any symptoms are produced. So this process of primary and secondary immune response is known as natural active immunity. So you can see from the graph here that the concentration of antibodies in the primary immune response is relatively low, and it takes a long time for these antibodies to be produced. However, when we're exposed secondary time round to an antigen that's non-self, the concentration of antibodies rapidly increases, and this occurs very efficiently in the secondary immune response. So cell-mediated immunity. So we're just going to discuss the process of this. So it begins by a pathogen with antigens on its surface being present in the body, and this pathogen is then engulfed and digested by a macrophage in phagocytosis. So then, as we know, the macrophage presents the antigen on its surface and becomes an antigen-presenting cell. So then receptors on some specific T helper cells fit the antigens, they complementary bind, and the T helper cells are then activated and produce interleukins, which we know stimulate more cells to divide rapidly in order to form clones of activated T helper cells which can bind to a particular pathogen. So we've got a clone of active T memory cells in order to work during the secondary immune response if we're infected again, and we also have a clone of active T helper cells. So the cloned T cells are involved in a variety of different things. So they can develop into T memory cells, as I said. They produce interleukins, which stimulate phagocytosis and B cells to divide. And they can also stimulate development of T killer cells, which we know are involved in destroying infected cells and pathogens. So humoral immunity involves the body responding to antigens found on the outside of cells and then producing antibodies, which are soluble in blood and tissue fluid, and antibodies aren't attached to cells. So B cells have antibodies on their cell surface membrane, so for example immunoglobulin M, and there are millions of different types of B cells with different antibodies. So the process begins with a pathogen entering the body carrying the specific antigen or producing toxins which act as antigens. We then have the B cell with complementary antibodies binding to these antigens. And then the B cell engulfs and processes the antigens to become an antigen-presenting cell. So activated T helper cells then bind to this antigen-presenting B cell, and this what causes clonal selection. So the B cell with complementary antibody to the antigen is selected for cloning. So interleukins produced by these activated T helper cells activate the B cells. And these activated B cells divide by mitosis to give clones of plasma cells and B memory cells, and this is known as clonal expansion. So now we're going to talk a little bit more about autoimmune diseases. So autoimmune diseases occur when the immune system stops recognizing self-antigens and attacks healthy tissues. So when T regulator cells don't work effectively. So this can be due to genetics in families, and it causes the immune system to respond abnormally to a mild pathogen or to normal body microbes. So there are approximately 80 identified autoimmune diseases, and these can cause chronic inflammation and also complete breakdown of healthy tissue. One of the ways to treat autoimmune diseases is by immunosuppressant drugs, but the issue with these is that they can deprive the individual of natural body defenses against disease, so they're more susceptible to infection. So now we're going to move on to some questions based on that information. I've grouped the questions based on how similar they are so that we don't spend too much time going over specific questions. But first we're going to begin with antibody structure. So this question is, which letter identifies the antibody region that allows the distance between antibody binding sites to vary? Now remember, if you want to pause the video and try to work this question out for yourself before I go over the answer, then please do. So we need to identify which letter is showing the region that allows distance between antibody binding sites to vary. So we know that the antibody consists of a constant region, a variable region, and a hinge region. So if you remember back to the other slide, it's the hinge region which allows the distance between antibody binding sites to vary. So the hinge region allows flexibility, and the hinge region is where the letter C is pointing to. And so therefore our answer is C. So we can identify that A is the variable region, B is the disulfide bridge, and D is the constant region. Question 2. Antibodies are defensive protein carried in the blood. Complete the simplified incomplete structure of an antibody. Draw and label the missing parts on the diagram. So we can see that this diagram is missing our light chains, our polypeptide light chains. So therefore we need to draw those on. So I'm just going to show the answer here. So for this question, you actually needed a minimum of one light chain drawn on the outside of the heavy chain, but obviously we know that there should be two, and these should be labelled light polypeptide chains, although the mark scheme does allow you to label them as the variable region because they're obviously part of the variable region, and also antigen binding sites. Question 3. Which numbers labelled on the diagram represent the part of the antibody that has the same sequence of amino acids in all antibodies? So we know that it's in fact the constant region of antibodies that remains the same. So we need to identify which numbers represent the constant region. So if we have a look at the diagram, we can see that number 1 is pointing to the top of this chain. So if you have a look at this marker here, we can see that this part is part of the variable region because this is what changes based on the specific antigen. So therefore, we can identify that 2 and 3 are actually part of the constant region. So therefore, our answer is C. Question 4. Name and describe the function of the parts of the antibody labelled X, Y and Z. Now this antibody looks a little bit different to the structures we've looked at previously, but don't let this faze you. So first we have X. So we know that X is in fact identifying the variable region, and this is the region where the specific antigen binds to the antibody. We then have Z, so Z here is identifying the hinge region, and this is what allows flexibility within the antibody. We then have Y, so Y is the constant region, so this remains the same for all antibodies, and I haven't discussed this previously, but the constant region is also involved in allowing the antibody to bind to phagocytes, so you can see our answer here. Question 5. Which of the statements about the labelled antibody are correct? So we have several statements here, and we also have an antibody diagram. So first we have X, which is here. So we know that the bonds within the antibody are disulfide bonds. So if we have a quick look over to the statements, we can see that C and D state that X is a hydrogen bond and that X is an ionic bond. So without even reading the rest of those statements, we can discount C and D, which was pretty easy because now we've only got 2 left. So then we have Y. Now I think this question is a little bit tricky because it doesn't, in the diagram of the antibody, it doesn't put the 2 little lines here to identify the variable region and the constant region. So if we just put them in there, so and the way that they've labelled Y, it could be misinterpreted as part of the variable region, but because it is slightly lower down, it is in fact part of the constant region. And then we have Z. So Z is, as we know, of course, part of the constant region, as this part is the constant region. So we know that Y and Z are the constant region and X is our disulfide bond. So if we have a look at A and B, A states that Y is the constant region and Z is a variable region, but B states that Y and Z are constant regions. So we know that B is our correct answer, and we can see our answer here. Question 6. So I'm not going to read through this whole question because it is a long one and this video is already very long, but basically we need to discuss celiac disease and why some people that stop eating foods that contain gluten still occasionally experience the symptoms of celiac disease. So if you want to pause the video and have a read through this question yourself, please feel free. So the questions are, what can we conclude about the structure of the antibody that causes celiac disease and what the antibody binds to when producing the symptoms of celiac disease? So despite the person not eating gluten, we could say that other foods may have a similar antigen. And remember that an antigen is a short sequence of amino acids. So then about the antibody, we can state that the variable region or the binding site is not specific to gliadin antigens. So the antibody binds to a T lymphocyte or a mast cell, and then this mast cell releases histamine, causing inflammation. Question 7, select the statement that describes an autoimmune disease. So we've got A, an individual's own body cells are antigenic, B, a pathogen attacks cells of the immune system, C, antibody production is prevented, or D, an individual has developed immunity. So we know that D is not an autoimmune disease because autoimmune diseases are bad and we develop immunity via primary secondary immune response or a vaccine, so D is incorrect. C then says antibody production is prevented. So autoimmune diseases don't prevent antibodies being produced, but they do in fact cause antibodies to be produced to kill our own body cells. And then B, a pathogen attacks cells of the immune system. So this is just a normal disease or infection. So this is not an autoimmune disease. So therefore our answer is A. So an individual's own body cells are antigenic. So here we can see our answer. So an autoimmune disease, is when the immune system stops recognizing self-antigens and attacks healthy tissue. So what the statement means here is by our own body cells being antigenic is that they are being identified as non-self antigens. Question 8. The figure shows the new antibody concentration in the blood of a person infected by a pathogen for the first time, so their primary response. On day 30, this person was infected with the same pathogen. Sketch a line to show the antibody concentration from day 30 onwards. So we know that if the person infected with the same pathogen, this is the secondary immune response. So the secondary immune response is more rapid, so it takes a shorter amount of time to produce a higher concentration of antibodies. So our sketch is going to look something like this, and our answer can be shown here. So questions 9, 10, 11 are all very similar. They are all about the primary and non-specific defense mechanisms. So the first one is which of the options is a primary defense mechanism against pathogens. So we have neutralization, agglutination, phagocytosis, or blood clotting. So we know that our answer is therefore D, because agglutination and neutralization are part of the specific immune response, and phagocytosis is part of the non-specific, whereas blood clotting is a primary defense mechanism. And we can see our answer here. We then have our next question. So A, antibody production, B, inflammation, C, phagocytosis, or D, T killer cells. So another primary defense is, of course, inflammation, which is B. So antibody production is specific, phagocytosis is part of the non-specific response, and T killer cells are obviously involved in the specific response as well. So we can see our answer here. We then have question 11, which is the same question, but our answers are antibody molecules, the HPV vaccine, lysozyme, or penicillin. So we know, of course, that lysozyme is a primary defense against pathogens, and we can see our answer here. Question 12. Epithelial cells in respiratory tracts are essential for defense against pathogens. Explain the function of epithelial cells in the areas of mammals in defense against pathogens, and suggest the importance of the cytoskeleton in carrying out this function. So this is quite a detailed question, but it's asking us to explain the function of epithelial cells in defense and the importance of the cytoskeleton. So we know that epithelial cells produce goblet cells and mucus. So goblet cells are what secrete and produce this mucus, and the mucus itself is what traps pathogens. So then epithelial cells are also involved in phagocytosis. So neutrophils, macrophages, and lysozyme are also produced in order to defend against pathogens and for the epithelial cells to regrow. And then we have cilia as well. So cilia are involved in wafting the mucus or beating it up out of the system to be coughed out or swallowed into the stomach to be destroyed. And the cytoskeleton's role is to move or to make up these cilia. And you can see our answer here. Question 13, describe the inflammatory response. So we know the inflammatory response is triggered by damaged cells or by pathogen presence, and then mast cells release histamine. Histamine causes vasodilation, so increases blood flow to the specific area of infection, and it also increases the capillary wall permeability, which makes it easy for phagocytes to leave the blood and enter the infected tissue. And we can see our answer here. Question 14. Betinin is a pigment found in beetroot that is shown to interfere with the action of cytokines acting as anti-inflammatory agents by interfering with cell signaling cascades that initiate, regulate, and amplify the inflammatory response. Suggest how betalain pigments could interfere with the actions of cytokines. So the way that they interfere could be that they prevent the cytokines from binding to cell surface receptors of target cells. So if the betinin bind to cytokines or the cell surface receptors, this means they are competing with them for the receptor binding site. And you can see our answer here. Question 15, describe one specific role of cytokines in the immune system. So we've discussed a few roles of cytokines, such as them stimulating B cells or T cells to differentiate, and also stimulating B cells to release antibodies. They also inhibit virus replication and activate T killer cells, and something else that they're involved in is chemotaxis, as well as the attraction of neutrophils to the site of infection. So you can see our answers here. Question 16, which of the following antibodies increases the phagocytosis of pathogens? So we have opsonins, agglutinins, and anti-toxins. So we know that Antibodies act as opsonins and agglutinins to increase phagocytosis and cause cells to be engulfed. However, when they act as antitoxins, it's because they counteract the toxins that are produced by pathogens. So therefore our answer is B. If you have a look back at slide 26, remember I will upload this PowerPoint to my website soon, and you can also flick back in the video of course as well. But antibodies act as opsonins, so the antibody complex is easily engulfed and digested by phagocytes. They act as agglutinins, which allow phagocytes to engulf several pathogens. They act as antitoxins, which means binding to toxins produced by pathogens, making them harmless. Question 17, explain what the terms opsonin and phagocytosis mean. So we know that opsonins are antibodies that enhance phagocytosis by marking or tagging antigens for easy identification and destruction. So then phagocytosis is the process by which the phagocyte engulfs pathogens. And we can see our answer here. Question 18. Phagocytes defend the body by engulfing and destroying pathogens during phagocytosis. Describe the 2 corrections that should be made to the summary of phagocytosis shown. So I'll allow you to pause the video and read through those stages, but I'm just going to go through the answers here. So stage 2, which says phagocytes recognize pathogen as self, should read phagocytes recognize pathogens as non-self, and then stage 4 and stage 5 should be the other way around. So stage 5 should come before stage 4, or stage 4 should come after stage 5, and you can see our answer here. Question 19, which of the structures labelled A to D in the diagram is a neutrophil? So here we need to remember the structures of the specific white blood cells. So neutrophils, we know, have a lobed nucleus and granules, so we can therefore identify the neutrophil as B, and we can see our answer here. So A is a monocyte because it's large and it has a kidney-shaped nucleus. C is a lymphocyte because it's slightly smaller and it has a nucleus which fills most of the cell. And then D is of course a red blood cell which has the characteristic curved dome shape. Question 20. The photograph shows a blood smear. Which row correctly lists the cells that are visible in the smear? So if we have a look at the photo, we can see that these here are of course red blood cells, and then which are erythrocytes. So red blood cells are also known as erythrocytes, and we don't seem to have any cells with a nucleus that fills the whole cell, and there aren't any monocytes either, but we can identify these cells as neutrophils due to this disjointed lobe nucleus, and you can see our answer here. Question 21, so another similar question about identifying the cells. So we have X here, Y and Z. So X is evidently a neutrophil because it has again the disjointed lobe nucleus. Z is a lymphocyte because it has the nucleus taking up most of the cell, and Y is therefore a monocyte because it's a larger cell with a kidney-shaped nucleus. So therefore our answer is C, and you can see our answer here. Question 22, which of the leukocytes pictured can differentiate into a plasma cell during the specific immune response? So a leukocyte is another term for a white blood cell, so don't get confused between leukocyte and lymphocyte. So leukocyte is a name for a white blood cell. So it's asked us to identify which picture shows the cell that differentiates into plasma cells. So we know that it's B lymphocytes that differentiate into plasma cells. So if we have a look at the cells, we can see that our answer is going to be D because in lymphocytes the nucleus takes up most of the cell. So you can see our answer and explanation there. Question 23, so we've got to name the cells G and H. So we can see that G has another lobe nucleus, and H, the nucleus, is filling up most of the cell as well. So therefore G is a neutrophil and H is a lymphocyte. Question 24, compare the roles of B and T lymphocytes in the specific immune response. And this is 6 marks, and it also has an asterisk, which means this is level mark, so it needs to be written clearly with key scientific terms. So when we say compare as well, this key word means that we need to identify the similarities and the differences. So the similarities between B and T cells is obviously that they both have complementary receptors, they both differentiate, they both have memory cells, specificity, clonal selection and expansion. And then for the differences, B lymphocytes are obviously processed in the bone marrow, whereas T cells are processed in the thymus. B lymphocytes have specific antibody production and differentiation into plasma cells. We can also talk about T helper cells, T killer, T regulatory T cells, as well as the use of cytokines and the stimulation of B lymphocytes. So these are some of the key terms here. So these need to be written in clear and concise sentences explaining the full process. Question 25, describe the process that lead to production of antibodies against invading pathogens. So we of course know that it's B cell differentiation into plasma cells which produce antibodies, but first we need to say that the B cells have antibodies on their surface which are complementary to a specific antigen, and then this activated B cell proliferates or divides and then differentiates into effector cells which and then differentiate into plasma cells, and it's these plasma cells which secrete antibodies which are specific to an antigen, and you can see our answer here. Question 26, explain how the processes that lead to the production of antibodies against invading pathogens are increased in speed by T helper cells. So we know that T helper cells are stimulated by antigen presenting cells, and when they're stimulated they then release cytokines and interleukins, specifically interleukin 2. These chemicals that they release then stimulate the B cell, which causes proliferation or clonal expansion. You can see our answer here. Question 27, explain why toxins produced by different strains of bacteria are immunologically distinct and how the immune system tackles the invading pathogens. So again, this is a 6 marker and it has an asterisk. That means it needs to be clear and concise. It's level marked, needs to have clear sentences and specific scientific terms. So I'm not going to go through the full answer here, but basically in terms of the toxins being immunologically distinct, you need to talk about specificity and then about how the immune system tackles invading pathogens. This is obviously talking about the specific immune response and how T helper cells stimulate B cells differentiate into plasma cells which produce antibodies. But you can see all of the key points here and how they interlink and how you can create a good answer. Question 28, statements A to F summarise the different stages of an immune response to a virus. Place the letters representing the statements in the correct order. So without even looking at the statements, we know that the first stage of the immune response to a virus is being engulfed and digested in phagocytosis. So therefore F is going to be our first statement. We then know that the macrophage presents the viral antigens on its surface. So C is our next statement. And then after this, T helper cells are activated and divide by mitosis, so then A, and the activation of these T helper cells causes them to release cytokines. So then we have D. Cytokines then stimulate B cells which become activated, divide by mitosis and then differentiate. So B is our next statement. And finally, we know that when B cells differentiate, they differentiate into effector cells, which then differentiate into plasma cells. And these plasma cells synthesize and release antigen-specific antibodies. So therefore, our final statement is E, and you can see our answer here. Question 29, describe the role of T killer cells in the immune response to HPV. So the role of T killer cells in the immune response is to kill or destroy infected cells. And then finally, question 30, if you've made it this far, then well done. Antibody levels were measured after 2 weeks after the first dose of vaccine. They were significantly lower than those measured after the second and third doses of vaccine. Explain why. So this is asking us to discuss the primary and secondary immune responses. So the first exposure to the pathogen, or in this case, because it's a vaccine, it'll be an attenuated pathogen, which means it's non-viral, it's not going to harm you as such. But anyway, the first exposure, so there's not going to be any memory B cells produced, so this means it takes time for clonal selection and expansion and for plasma cells to be produced, and only a small number of plasma cells are produced. But by the second and third exposure, the memory cells are present, they are stimulated to divide rapidly, and so clonal selection and clonal expansion are also faster. So this means that the memory cells differentiate into many plasma cells and they produce antibodies faster and at a greater concentration or in greater numbers. And you can see our answer here. So much for watching. I know this has been a really long video. There was a lot of questions on immunology for OCR, but I hope that by me including all these questions, this is something that's really helpful for you and useful to look back on when you're doing revision and questions on immunology. Make sure to follow my social media and also to give this video a big thumbs up if it has been useful for you, and please subscribe to my channel. We are two subscribers away from 200 subscribers, which is really exciting, so please subscribe. But thank you again for watching, and I'll see you in my next video. Bye guys.